Hey guys, and welcome back. So today we're going to be talking about animal cells and plant cells, and basically kind of talking about the differences, the similarity and differences between animal cells and plant cells, and talking about the cell structure in terms of the organelles that are in animal cells and plant cells, and talk about their function and structure, everything like that. So let's get started. So both animal cells and plant cells are eukaryotic cells, and eukaryotic cells are basically cells that contain a nucleus. And although plant cells and animal cells are eukaryotic cells, they do share differences, similarities and differences in their cell structure. So for an example, animal cells, they have centrioles, whereas plant cells do not have a centriole, but at the same time, both cells contain a mitochondria. And if you are studying cell structure and anything like this in the exam, then it's very important for you guys to know the cell structure, the function, and everything to do with it, because especially when I'm talking about GCSE and A-level exams, they're going to ask you about it, and they will link cells, and they may even link cell structure to different parts of the topic of biology, in different parts of topics in biology. So it's very important for you guys to know that. So the first one I'm going to talk about is the centrioles. So animal cells have centrioles, plant cells do not. And these centrioles, they basically consist of two bundles of microtubules, which is a type of fiber in the cytoskeleton, and they are perpendicular at right angles, 90 degrees, whatever you like to say, to each other. And these microtubules are arranged in a 9+2 arrangement, which is basically where the 9 microtubules are arranged into a circle, and the 2 microtubules are kind of like positioned in the center. The centrioles are needed for cell division, mitosis, which I'm sure maybe some of you guys may know that. And the centrioles are needed because that is the formation, because that is where spindle fibers are formed. As you may know from mitosis, the spindle fibers are the ones that attach to the chromosomes, which are aligned in the equator or in the middle of the cell, and then they move them apart from each other, split the chromosomes apart from each other to either end of the cell, to the opposite poles of the cells. Yeah, that's it. That's not it, because the centrioles is also used to form cilia and the flagella. Yeah. And basically the microtubules would kind of extend out of a cell's plasma membrane to form these cilia and flagella. But also, even though I did say that plant cells do not have a centrioles, but there are exceptions. There are, some unicellular green algae plants may have a centriole, but usually when we're talking in general terms, in terms of plant cells, they generally do not have a centriole. What I'm going to talk about is mitochondria. So both animal and plant cells have a mitochondria, and they are normally, like, when people describe the role of the mitochondria, they kind of say it's the powerhouse of the cell, which basically means that, they release energy, and the mitochondria is also the site of AT production during aerobic respiration. So normally in exam, if they were ever going to ask you the function of the mitochondria, then it's normally like production of ATP, or the site of aerobic respiration, which is used to release energy. These organelles, so mitochondria, are self-replicating since they have their own DNA. So if there's an increased demand for energy, then they can just replicate themselves and create more mitochondria. Cells such as muscle cells, liver cells, which has a high metabolic activity, contain lots of mitochondria. And in terms of their structure, they're kind of spherical. Well, it depends on the way they cut the cell. So sometimes you may see in an exam that it will be like a circle, or sometimes they may be like a kind of like a sausage shape. It is double membraned, so has double membraned, and the inner membrane is highly folded into a cristae. And in the mitochondria, why am I mixing up in a mitochondrion, there is a fluid filled, there's kind of like a fluid in there, and that's basically called the matrix. Both animal and plant cells have a plasma membrane, but I want to talk about that in another video because there's a lot to say about the plasma membrane. So yeah, see you then. We also have a rough endoplasmic reticulum, which is also known as the RER, and this is an intracellular transport system. So the RER, it has a cisternae, like it has like lots of cisternaes, and which is a fluid filled cavity, and they basically are used to transport substances from one area to another. I'm sure the key thing about an RER, the rough endoplasmic reticulum, is that they contain ribosomes along their surfaces, and that basically provides a larger surface area for the ribosome so that amino acids can attach to each other. And the ribosome is known for protein synthesis. That's where you do protein synthesis in the ribosome when you're making proteins. And the protein kind of travels through the RER and is then transported to the Golgi apparatus for further modification and packaging. The smooth endoplasmic reticulum, also known as the SER, has kind of the same structure as the RER. It has a cisternae, which is the fluid-filled cavity, but a main difference is that it does not contain any ribosomes. In the exam, when they are asking you about the function of a smooth endoplasmic reticulum, you should say synthesis of lipids. That is literally it. Cholesterol, you have cholesterol. Oh, what other lipids are there? Some steroid hormones. But if the exam ever asks you what is the function, what is the role of the SER, of the smooth endoplasmic reticulum, always put synthesis of lipids. It's synthesized lipids, basically. Cytoplasm is basically a jelly-like substance, which is where chemical reactions take place, and organelles are also suspended in the cytoplasm as well. The Golgi apparatus modifies and packages the protein into vesicles so that it can be transported to the plasma membrane and secreted by exocytosis. You will know more about that in a later video, which will be linked with my plasma membrane video, just to let you know. Or it can just be stored, all the protein in the vesicle can just be stored in the cell. Cytoskeleton is just a network of fibers that run throughout the cell. So there are 3 types of fibers in the cytoskeleton. So you have the microtubules, you have the intermediate filaments, and you also have the microfilaments. And the cytoskeleton overall kind of provides the cell support and gives the cell its shape and structure. It is also used in transport. So like there are proteins on the microtubules that move up and down the microtubule while carrying a vesicle which contains proteins that is needed to be transported to the Golgi apparatus and the plasma membrane. And these proteins that move up and down the microtubule are called protein motors, but I've also seen that they're also called microtubule motors. But sometimes it's on the safe side. I recommend you to put protein motors, but I'm sure examples will still give you credit if you say microtubule motors. And the cytoskeleton is also used to form the cilia and the flagella, which is used for cell movement, because the role of the flagella is to move the sperm cells, so to move. And for the cilia, it moves like this. Well, I'm imagining it moves like this. it creates somewhat of a, if there's lots of cilia together, then it creates a sort of current, and it basically moves the mucus, which contains trapped dust and microorganisms, basically away from the lungs or wherever it is. Away from the lungs, but they are also, sorry, this is kind of extra, but they also are the ciliated epithelial cells, which contain these cilia. They also move the egg cell, because the egg cell doesn't move by itself. The cilia on the ciliated epithelial cells move the egg cells. Both plant cells and animal cells contain a nucleus. That's why they're eukaryotic cells anyway. The nucleus contains DNA, which provides genetic information or genetic material, depends which one you like to say. And it is also used in protein synthesis to make proteins. It's used in the process of protein synthesis. The nucleus has a double membrane called a nuclear envelope, and the nuclear envelope contains like these nuclear pores, so they're basically holes, and this allows larger substances to pass through to like enter and leave the nucleus, such as messenger RNA, which is needed in protein synthesis, to leave the nucleus so they can go to the ribosomes, and some steroid hormones which enter the nucleus. The nucleolus contains RNA and doesn't have a membrane, and this is where the ribosome is made. Lysosomes, which both plant cells and animal cells do have, are formed from the Golgi apparatus and are small bags that contain hydrolytic or digestive enzymes. Phagocytes such as neutrophils contain lots of these lysosomes, because, as you may or may not know, phagocytes is a type of white blood cells, and it basically kind of surrounds and kind of engulf the pathogen, and these lysosomes, which contains these hydrolytic enzymes, kind of just digest because the main role of the white blood cell is to destroy pathogens or foreign organisms, microorganisms, whatever you like to call it. The hydrolytic enzymes in the lysosomes digest old cell organelles and foreign cells, which I've just said. Chloroplast is only present in plant cells, not animal cells. They are the site of photosynthesis and are large organelles. I have seen in a question, they were like, why can you see the chloroplast like in this diagram, in this figure, like in figure 1, which kind of shows like what you can see under a light microscope? And the answer to that is basically because they are large organelles. They are large. Just kind of like a side note, just to add that on. Similar to a mitochondria, because chloroplast contains a double membrane where the inner membrane contains stacks of flattened sacs called phycoloids. Each stack is called a granum, and chloroplast also contained a fluid-filled matrix, which is called the stroma. And similar to the mitochondria, chloroplasts have their own DNA. The cellulose cell wall provides strength and support for the plant cell by preventing the plant cell from bursting when turgid. It maintains the cell's shape, and the cellulose is permeable, allows solutions to enter and pass through the cell wall. Plant cells have a cellulose cell wall, whereas animal cells do not. Plant cells also contain a permanent vacuole, and the permanent vacuole contains a membrane called tonoplast, which surrounds the vacuole. Plant cells contain a large permanent vacuole, whereas an animal cell does not. Provides the cell stability and contains water and solutes. And this is because when the cell is turgid, the vacuole pushes against the cell wall. Thank you guys for watching. I hope you guys found that good. Make sure to like and subscribe if you like this video and you found it interesting and you found it good. So thank you guys, and bye. See you next time.